Good morning, students. Today we're going to learn about this next chapter in genetics and biotechnology, session 3, Mendelian principles. In the last class, we learned about heredity and environment, where we concentrated on norm of reaction, interaction between heredity and environment, experiments on potentilla glandulosa, and also related to fur color inheritance in Himalayan rabbit, and studies related to human twins. Today, we are going to concentrate on Gregor Mendel's experiment on pea plants, monohybrid cross, backcross, test cross, and the two laws which we are going to learn is law of dominance and law of segregation. Terminology: monohybrid cross, the cross between two organisms differing in a single pair of contrasting characters. That is, we take up two parents and which are differing, or we are concentrating on only one character at a time. For example, height of the plant or color of the eye, etc. Test cross refers to cross between the F1 with the recessive parent. Back cross refers to cross between the F1 and any one of the parents. It may be either dominant parent or the recessive parent. So there are two back cross 1 and back cross 2. F1 or the first filial generation. In hybridization experiment, when the parents differing in a pair of contrasting characters are hybridized, the first generation of organisms which results is called as F1 generation or first filial generation. And after selfing this F1 generation, the next generation which is produced is called as F2 or second filial generation. This is the great scientist in genetics who is called as the father of genetics, Gregor Mendel, 1822 to 1884. He was an Austrian monk, and he was father at Braun in Austria, then Austria, now it is called as Czechoslovakia. He conducted hybridization experiments with the pea plants, Botanigida. Scientific name is Pisum sativum, and he did his experiment for 8 years, and he discovered the fundamental laws of heredity, which is known as Mendelism. He grew over 10,000 pea plants, and he kept a track of the progeny number and type. He recognized the mathematical patterns of inheritance from one generation to the next generation, and he came out with a beautiful explanation which is called as Mendelian genetics or Mendelian laws, and which he presented in the annual proceedings of the Natural History Society of Braun in 1866. Mendel's work and his laws of inheritance were not appreciated at that time. Though it was a great discovery related to genetics, which laid the fundamental principles of genetics, nobody recognized it. But after his death in 1900, three different scientists from three different parts of Europe, Correns, De Vries, and Teschermark, they rediscovered his results, and everybody appreciated and understood the basic laws of genetics experiment which Mendel did. This is the pea plant, Pisum sativum, Botanigida. We can find out lot of variations in the characters. And I think Mendel, after careful studies, he selected 7 characters, contrasting characters, which he took for his hybridization experiments. I think you can make out the different uh, characters. One of the character is the presence of the flower at the axis, presence of the flower at the axis or terminal flower or axial flower, height of the plant. This is the tall plant, and this is the short or the dwarf plant. The character is, or the trait is, height of the plant. In relation to height of the plant, two contrasting characters he identified. One is the tall plant and the other one is the dwarf plant. Like that, he carefully selected 7. He observed, though he observed 36 variations, he carefully selected 7 contrasting variations, which are as follows: stem length, flower position, whether it's axial or terminal, flower color, purple or white, pod color, uh, green or yellow, pod shape, inflated or constricted, ಉಬ್ಬಿರೋದು ಅಥವಾ ಸೊಟ್ಟೆ ಆಗಿರೋದು, seed form, round or smooth. This refers to the seed which is present inside the pod. I think when we open the pod, we can find out the seed, which is generally smooth or wrinkled, generally smooth, rarely wrinkled, cotyledon color. That is, inside the seed, cotyledons will be there, which are yellow or white. It's easy, I think, if you remember, ಅವರೆಕಾಯಿ ನೋಡಿದಾಗ ಅವರೆಕಾಯಿ ಬಿಡಿಸೋವಾಗ ನಮಗೆ ಗೊತ್ತಾಗುತ್ತೆ ಅಲ್ಲ, ಒಳಗಡೆ ಗ್ರೀನ್ ಅಥವಾ ಯೆಲ್ಲೋ ಇರುತ್ತೆ. ಅದೇ ಥರನೇ ಇಲ್ಲಿ ಯೆಲ್ಲೋ ಆರ್ ವೈಟ್ ಇನ್ ರಿಲೇಷನ್ ಟು ಪೀ ಪ್ಲ್ಯಾಂಟ್ ಆರ್ ಬಟಾಣಿ ಗಿಡ. These are the characters, pictorial representation of the same Mendelian characters. The dominant characters are shown in the below line. That is the color. First one is the color of the flower, and the last one is the height of the plant. Tall is dominant over dwarf, and in purple and white, purple flower is dominant over white. Purple flower, and this is the tall one, and axial flower is dominant. Then yellow color of the seed is dominant over green. Then round is dominant over wrinkled. Pot shape, that is inflated, is dominant over constricted. Green pod is dominant over yellow pod. Monohybrid cross. The cross between two parents differing in a single pair of contrasting characters is known as monohybrid cross. See, any organism is a bundle of characteristics wherein we find hundreds or thousands of characters. But we cannot study all the characters at a time. At a time, for the experiment or for the observation, only one character is taken. And when we take a single pair of contrasting characters, and when the hybridization experiment is carried out, that is called as monohybrid cross. Resulting individual is called as monohybrid. Example, a cross between a homozygous tall represented by genotype T, and with homozygous dwarf, genotype is small t, pea plants. A cross is carried out between a homozygous tall pea plant with a homozygous dwarf pea plant, genotype being T and small t for the dwarf plant. That cross between tall and dwarf plant, which are homozygous, results in the production of tall plants in F1. In F1, all the plants produced will be tall and they are heterozygous. That is, the genotype is T and small t. And when selfing of the F1 takes place, that is, when F1 is hybridized among themselves, this is called as selfing of F1, it results in F2 plants in the next generation, where the approximate ratio of tall is to dwarf plant is nearly 3:1. That is, 3 tall plants and 1 dwarf plant is produced phenotypically, and the genotypic ratio is 1:2:1. 1 is T, that is homozygous for dominant character, and 2 is heterozygous for dominant, that is tall, T and small t, and remaining 1 ratio is recessive trait, that is small t and small t. T and small t are alleles of a gene which influence the height of the plant, and in this experiment, tall is dominant over the dwarf. The same thing is expressed here. This is how the genetic cross are written. Mendel crossed pure breeding tall pea plant with the dwarf pea plant. Here you can make out the parent generation. Parent generation. The phenotype is tall pea plant and the with a dwarf pea plant. Both are homozygous. So the genotype is capital T for tall, and for dwarf it is small t and small t. When these plants produce the gametes, meiosis reduction division takes place. So only one of the alleles will enter into the gametes in relation to height of the plant. So the tall plant produces a single type of gametes having capital T in it, and the dwarf plant produces small gametes which have small t allele in the gametes. Then when they fuse, when they undergo fertilization, it results in the production of F1 plants, and all the F1 plants will be tall with a genotype capital T and small t. Then selfing of the F1, that is, this is heterozygous tall plants. When the selfing of the F1 plant is undertaken, each plant, F1 plant, produces two types of gametes. That is, 50% of the gametes produced will have capital T allele, and another 50% will have small t. So this when the selfing of the F1 takes place, random fusion takes place, it results in F2, wherein we get four different types of offsprings which are capital T is to 2 capital T and small t, then small t, 1 small t, 1 is to 2 is to 1. Capital T are homozygous tall plants. Capital T and small t are heterozygous tall plants, and small t are homozygous dwarf plant. The same thing is represented in the checkerboard method. Here you can, in this side, you can make out the hybridization experiment which we just learnt. That is, this is the parental generation, wherein a tall pea plant is crossed with a dwarf pea plant, resulting in F1, or the F1 are heterozygous for tall, where they have capital T and small t genotype. Then when the selfing of among F1 takes place, it results in four different types of plants. One capital T, that is homozygous tall, 2 heterozygous tall, and 1 homozygous dwarf. That is, selfing of F1 plant is also represented using the checkerboard method, wherein the female gametes are written on x-axis and male gametes are written on the y-axis, wherein you can make out. That is, by filling the checkerboard, we can get the probable genotypes among the F2, where there is 1 homozygous tall, 2 heterozygous tall, and then 1 homozygous dwarf. The same thing is represented in the checkerboard method. Then the second cross is this character is color of the flower. In this case, the parent is having that is, one of the parent is homozygous for purple flower, and the other parent has the white flower. Then they both are homozygous. Then when they are crossed with each other, they produce F1. All the F1 have purple color, and it is heterozygous purple, capital P and small p. Now, when selfing of the F1 takes place, that results in F2, wherein we get 3:1 ratio of the flower, 3 purple color flowers and 1 white colored flowers. See, for example, there are If there are 400 plants produced, then out of that 299 or 298 plants will bear the purple flower and remaining flowers will bear the white flowers. So in this, among the purple flowers, one is homozygous tall and the other two are heterozygous tall, capital P and small p, and the last one is homozygous for white color, that is small p and small p. The phenotype of this tall plant is, purple plant is capital P and capital P, homozygous for tall. This is heterozygous for this is homozygous for purple flower, capital P and capital P. This is heterozygous for purple, capital P and small p. This is also capital P, small p. And the last is the recessive alleles are present, that is small p and small p, which produce white flowers. This is the second example. Next, I think you can do this. Carry out the monohybrid cross between pure lines of plants with round seeds and wrinkled seeds. Round seed are dominant over wrinkled seeds. Find the phenotype and genotype of F1 and F2. Then coming to Mendel formulated certain assumptions based on his experiment, and these are put together as laws of dominance. Second one is law of segregation or law of purity of gametes, which both are related to monohybrid cross. And the next law is law of independent assortment, which is related to dihybrid cross, which we are going to learn in the next chapter. Law of dominance. Some will not consider this as the true law of Mendel. Law of dominance, some scientists will not consider this as one of the laws of Mendel, but whereas some consider this as the first law. Either way, it doesn't make much difference. The law of dominance, that is, we understood a few examples related to monohybrid cross. In the cross between homozygous organisms for a contrasting characters of a pair, only one of the character of the pair appears in the first filial generation. The character which appears in the F1 is called as dominant character, and it conceals or suppresses another character, that is the recessive character. In the first example, I think you remember, the capital T suppresses the expression of the small t, which is the dwarf character. So in this case, capital T is dominant, that is the tall allele is dominant over the dwarf allele small t. And in relation to purple and white flower, capital B or capital P, either of the alphabets we can use. Capital B purple allele is dominant over small allele, that is small b, which stands for white character, white flower. Then we need to learn about monohybrid test cross and back cross. When the test cross is the cross between F1 and the recessive parent, back cross is the cross between F1 and any one of the parents. There are two parents, one is the dominant, other one is the recessive. So there will be back cross 1 with the dominant parent and back cross 2 with the recessive parent. Back cross 2 is almost similar to test cross. So this is the test cross, where the cross is carried out between the F1 and with the recessive parent. The genotype is small b for white flower color. Then purple color, because it is F1, it is heterozygous, capital B and small b, which produce, this plant will produce two types of gametes, 1:1 ratio, capital B and small b. And then the recessive parent will produce only one type of gametes, wherein all the gametes will have only one allele, that is the small b. When random fusion takes place, it results in the production of 1:1 purple and white flowers, the genotype being capital B, small b, and white flower has small b and small b. Recessive character, no, it is invariably it will be having small b only. Then only it can have the white flower. Then backcross 1 is with the F1 parent. Backcross 1 is with the F1 parent with a purple flower, wherein the genotype is capital B and small b, with the dominant parent, that is the pea plant with purple flower, represented by genotype capital B and capital B. It produces only one type of gametes, wherein all the gametes will have capital B in it. And the F1 plant will produce two types of gametes, 1:1 ratio. 50% of the gametes produced will be having capital B, and another 50% will have small b. Then when the fusion takes place, when the fertilization takes place, all the plants produced will have the purple flower. And the genotype of this purple flower is 1:1 ratio of capital dominant homozygous purple flower with heterozygous purple flower, wherein capital B and capital B small b. Monohybrid backcross is similar to the test cross, where the F1 is crossed with the recessive parent. Here you can make out the genotype of the F1 is capital B small b, because it is heterozygous. They produce two types of gametes, and the pea plant with white flower produce only one type of gametes, because it's a recessive alleles are present together. Then when the fertilization takes place, it results in 1:1 ratio. One purple is to one white. Genotypes are shown there. This is heterozygous for purple and homozygous for white. Law of segregation. In F1, two contrasting alleles, that is in the previous example, capital B for purple and small b for white, they are brought together in a monohybrid cross. But during gamete formation in F1, the alleles are separated or segregated in pure form without any contamination or blending. Thus, the purity of gametes is maintained. That is, see, when the parents undergo hybridization, the capital B and small b will come together and stay for one generation in F1 plant. In spite of staying together for one complete generation in F1 plants, they do not undergo any contamination or blending or mixing. So, when the F1 plant produce the gametes, the alleles capital B and small b, they segregate completely in the pure form as two types of gametes in 1:1 ratio. Half of the gametes will have capital B and another half will have the allele or the gene for white flower, that is small b. So this is the law related to segregation, law of segregation or law of purity of gametes, where the gametes or the alleles will maintained in the pure form in those staying together for one complete generation. You can do these problems related to monohybrid cross. In human beings, brown eye is dominant over blue eye. A brown-eyed man marries a blue-eyed woman. All their children are brown-eyed. Show the genotypes of the parents and children. See, a brown eye means it's a dominant character. So the possibility is it may be homozygous brown or heterozygous brown. You need to find out what are the possibility if the man is homozygous for brown or the heterozygous for brown, and which one suits in this case. Next question is, in Drosophila, a cross between a vestigial wing. Vestigial wing is a recessive character over the long wing, that is the dominant character with the long wing. They produce 20 offsprings. All the offsprings have long wing. What is the probability of this happening if the long wing fly is homozygous, capital L and capital L genotype, or heterozygous, capital L and small l? You need to carry out the cross and find the results. Outcome of this session is it helps students develop the conceptual understanding of the Mendelian genetics, monohybrid cross, and loss of dominance and law of segregation. For the Mendelian genetics, you can refer any genetic book wherein all these details are given, and you can refer Genetics by Dr. Veerapala Rastogi, and image sources I have written here in the reference. Okay. Thank you.